Okay, well, let's go ahead and get started. Aloha, and thank you everyone for coming to our 2026 Summer Undergraduate Research Fellowship Final Symposium, hosted by Hawaii Sea Grant College Program. We will start with a few words of welcome from our director, Darren Lerner, Dr. Darren Lerner.
You think she meant that literally? Get up and say a few words. No. Oh, no. Thanks, Rachel. Hey, thanks everybody for coming. I don't know. I can't see if we have how many we have online, and they couldn't hear me until now. Hi, thanks everybody for joining online and in person. We're always this is like one of the best things we do because we're what they call us a non-academic program. We're an organized research unit. And because we are that, we don't have a degree program. And so we have to work a little harder to connect with undergraduate students in particular. And this is one of the very best opportunities that we have to do so. And in addition, we are always, and we could have a discussion, or I bet you're not going to argue with me, but we think the best way to learn is through experiential learning. And I think certainly our students would all agree that this summer's experience is exactly that, at least when compared to our traditional kind of classroom learning, right? Sitting in a seat and listening to someone talk about something. So congratulations to all the students who made it through the summer and completed their projects. We're really super excited for you, and I'm looking forward to hearing the presentations today. So thanks.
And thank you, Darren, for being the man with the money. We have had a great summer. Of course. We have had a great summer with our cohort of 8 students getting together from 2 different islands for a couple of workshops. We had one student who survived a typhoon in Guam, and we had an amazing group of mentors. Most of whom actually spontaneously volunteered to be mentors, which is fabulous because then I don't have to chase after them. So we thank you very much for your willingness to give this great experience to the students. We could not do this program without you. So we are excited to hear the presentations today about all the great science that got done this summer and what the students themselves have actually been doing. So to keep things rolling though through the presentations, we're gonna limit to 1 or 2 questions after each presentation. So let's go ahead and get started. Hannah is our first speaker. So Hannah, why don't you come on up? Oh, there we go. There's Hannah. You get the clicker and you get a mic. So Hannah is a rising junior majoring in chemistry at UH Manoa. She's interested in water quality and helped to work on Kona Low water samples this summer with Andrea Kealoha in the Department of Oceanography. So you get to start us off, Hannah.
Yay.
Yay.
All right, well, my name is Hannah, and with my summer at SURF, I investigated the effects of the March 2026 Kona Low floods on coastal water quality on Oahu. The March 2026 Kona Lows consisted of 2 back-to-back low-pressure systems, the first one occurring from March 10th to 16th, and then again on March 19th to 24th. There's a cumulative rainfall of up to 76 inches, and the islands as a whole experienced a total of 2 trillion gallons of rain. The Department of Health issued island-wide brown water advisories for Kauai, Maui, Oahu, and then parts of Hawaii Island. And then if we look at our cumulative rainfall total from the Hawaii Climate Data Portal, we can see that from Oahu to Maui, they experienced about 16 to 32 inches of rain, some parts reaching up to that 76 inches. And then Kauai and Big Island experienced about 4 to 32 inches of rain. And then you can see some of that brown water that followed after the Kona floods. And then how do floods impact coastal water quality? Eutrophication is the over-enrichment of excess nutrients, typically nitrate and phosphate, and following a flood, there's short-term pulses of nutrients that stimulate the growth of phytoplankton and algal growth, which can trigger algal blooms. And then there you can see corals that are being consumed by excess algal growth. And then hypoxia is when a marine area experiences low dissolved oxygen levels, which is unable to sustain the typical marine life that's usually present. And then algal blooms often block sunlight, which can slow or stop photosynthesis. And then eventually bacteria will begin to break down the algal bloom with a process that completely consumes all available or most available oxygen. and then coastal acidification. So with the excess amount of phytoplankton and algae, there's going to be increased amounts of photosynthesis and therefore respiration, which also means an excess amount of carbon dioxide being released into surrounding waters, which will lead to lower pH levels. And then the floodwaters also introduce low alkalinity freshwater into the coastal waters, which lowers the coastal waters' ability to resist pH changes. And then sedimentation. Following floods, there's also going to be a large amount of suspended particles that cloud the water or make the water more turbid. And this will also slow down photosynthesis. And then eventually, when the suspended particles do settle, they bury benthic habitats and coral polyps. And then I also wanted to emphasize that these effects aren't just isolated events. They often happen simultaneously. They're triggered by one another. They can amplify each other. It's not just one event. I had 3 objectives throughout my investigation. The first one being to complete the laboratory analysis of coastal water samples for salinity, dissolved inorganic nutrients, and fluorescent dissolved organic matter. And then to investigate the coastal nutrient concentrations and the relationship to overall coastal water quality. And then to relate the observed nutrient conditions with the already observed impacts of elevated nutrient concentrations on coral reefs. What's in Your Water was a collaborative initiative by UH Manoa SOAS and Hawaii Sea Grant to launch a rapid response citizen science campaign to collect coastal water samples with the goal of addressing community concerns about floodwater impacts on coastal water quality. We prepared around 1,000 sample bottles in just 2 days, and then you could see one of our flyers that was circulated throughout Instagram and Facebook to try and engage with the community and get them involved with our sampling. And then you can also see an article that was done by the Hawaii Public Radio about this initiative, and I'm preparing hundreds of our sample bottles. We collected around 800 samples from this initiative across Oʻahu, Maui, Lanai, and Molokai with over 140 volunteers. We're hoping to measure 5 parameter groups from these samples with nutrients and salinity already being completed. We're currently in the process of measuring fluorescent dissolved organic matter, and then we hope to measure trace metals and stable isotopes, specifically delta 15N of nitrogen. And then in our top 2 photos, the one on the left, you can see a little bit more of that brown water. These were both taken by some of our sample volunteers. And then the one on the right, you can see one of our volunteers actually paddled out to get a sample that was a little bit further away from the coastline. And then we also have our What's in Your Water data portal. It has all of our samples that were collected along with their unique sample ID so that volunteers can go and look at the data that we collected from their specific sample. It has all of our updated data, all of the parameters, what they mean, how to interpret the data. And I think that this was the quickest goal of this initiative, was to just try and get data as quickly as we could to the community and as accessible as we could. That way they know how they were specifically affected and how they might be affected by floods and storms in the future. For my nutrient sample analysis, I use a SEAL AA500, which is a segmented flow analyzer. We have 5 different channels for nitrate, nitrite, phosphate, silicate, and ammonia. Nitrate is an indicator of agricultural and wastewater runoff. High levels often drive eutrophication and algal blooms, and it accumulates quickly from biological waste. Nitrate is an indicator of the active nitrogen cycle. It's actually the midpoint in that cycle, and it's a good indicator of when there's going to be spikes in nitrate concentrations. And then phosphate, like nitrate, is also an indicator of agricultural and wastewater. And then silicate is used to trace freshwater input, specifically groundwater. And ammonia indicates unoxidized organic waste from fecal matter and fertilizers. So to measure FDOM, or fluorescent dissolved organic matter, I used an aqualog or a spectrofluorometer. FDOM is often referred to as organic matter fingerprinting because it can be used to track the source or the composition of organic matter. And this data can be used to monitor how freshwater disperses into coastal waters. It can track terrestrial runoff as well as sewage and agricultural runoff. And then to measure salinity, we use a conductivity meter, which measures the concentration of dissolved salts in water, and it can be used to track freshwater input as well as monitor the balance between coastal and freshwater runoff, which is important because it helps us infer how healthy the ecosystem may be, water circulation, and any environmental stress or pollution. And then onto our data. Each of these dots on the map represents a specific sample, and then you can see the color represents the concentration of that sample. And this specific map is interpolated, meaning that basically a graph was drawn along the coastline, and using the nearest samples, we were able to hypothesize or calculate what the surrounding concentration might be. So looking at our salinity and nitrate data for the windward side, we can see that there are pretty wide stretches of typical coastal salinity values of 30 to 35 parts per thousand, but there are a few depressed salinity values along Waimanalo Bay, Kaneʻohe Bay, and Kahuku over on this map. And then when we look at the nitrate, we do see that there's a wide plume almost along the whole coast, but it's specifically, once again, concentrated around Waimanalo Bay, Kaneʻohe Bay, and Kahuku. When we compare the two together, we do see a correlation between depressed salinity values and elevated nitrate values, which is pretty typical following a flood or a storm. It just means that there's freshwater runoff that's carrying these nutrients into the coastal waters. And then looking at the North Shore, we see pretty typical coastal salinity values once again with 30, 35 parts per thousand, with a few depressed values around Pukea. And then looking at the nitrate values, we do see once again a plume almost along that whole north coast, concentrated around Haleiwa. And we'll be able to paint a better picture as to where it's coming from, but once we have our FDOM and our isotope data. But we do see a different correlation between salinity and nitrate, and it points to maybe a more localized access point. I was looking into maybe it being agricultural runoff from the old Wailua sugar plantation, but we also know there's a lot of cesspools that are concentrated in that area, so it could be cesspool runoff as well. And then looking out onto the South Shore, we see pretty typical coastal values along that shore as well, but with mildly elevated, but also inconsistent nitrate values, which would once again point to a more localized source. It could be submarine groundwater discharge. It could be cesspools as well. And then looking at the leeward side, we once again see pretty typical coastal values with a few depressions around Nanakuli. And then we see slightly elevated but consistent nitrate values on the leeward side with one elevated value around Nanakuli. And because there is once again that correlation between depressed salinity and elevated nitrate, it kind of points to a specific like freshwater runoff point. I know the Nanakuli Stream is right there, so it could be coming from there. And then how might coral reefs be responding to elevated nutrients? So I looked at a study previously conducted by Dr. Silberger, where the effects of elevated nitrate and phosphate were tested on coral reef calcification. There were 3 different concentrations tested: ambient, medium, and high. And during the medium concentration of 3.6 micromoles per liter, the corals did begin to share a more elevated response. We saw a decrease in net community calcification rates, meaning that the calcium carbonate being produced by the community as a whole decreased. And then the reef flipped from net calcification to net dissolution, meaning that the corals were dissolving faster than they were calcifying. And this could be attributed to 2 different factors, one of them being that the symbiotic relationship between zooxanthellae and corals was disrupted by the excess nutrients, as the corals could now get their energy from the nutrients rather than photosynthesizing. And another one being, as we talked about before, with excess nutrients and eutrophication, we also see an increase in stuff like algae, phytoplankton, and microbes. And these microbes attribute heavily to the coral exoskeleton's dissolution, meaning that with this increased population, it's likely that they were causing the coral to dissolve faster than they could recalcify. And then we also saw an increase in pH variability. As I mentioned before, with the increase in phytoplankton and algae, there's going to be increased photosynthesis and respiration, meaning an increased amount of carbon dioxide, which would lower the pH level. So that's why we chose the medium concentration levels to compare our data to. So if we look at the map, all of the red points signify a coastal site that exceeds the medium nutrient addition standards. We see it pretty heavily concentrated on the windward side, right along those same plumes in Waimanalo, Kaneʻohe, and Kahuku, and then we see it following that plume in Haleiwa as well. And then we also see it scattered about the South Shore, and then we see 1 or 2 points in Nanakuli area. And then looking at that same study with phosphate, the medium nutrient addition being 1.08 micromoles per liter this time. We still see it concentrated pretty heavily on the windward side, except not as many points by Kahuku. We see a few points on Pupukea and then 1 or 2 in Waikiki area. And the reason there might be less phosphate than nitrate exceeding these levels is because phosphate tends to latch on to soil particulates. So they move around less freely and not as fast as nitrate might. And then obviously, I think it's important to recognize that there are corals in these areas that are possibly being affected. We know there's spots in Nanakuli where the surface is covered 53% by corals, and then from Haleiwa down to the opposing windward side in Waimanalo, there is a steady area that's 50 to 75% surface level covered in coral reefs. And then my future work, I'd like to measure the stable isotope of delta 15N of nitrate for the viable what's in your water samples to complete source detection and figure out where this excess nitrate is coming from, whether it be agriculture or wastewater. And then I'd like to pair this study's conclusions with finalized FDOM data to build a more foundational understanding of FDOM nutrient correlations following the Kona Low storms, and hopefully they can be applicable to future storms and floods as well. And then I did go on a little side quest during my fellowship, and I got to go on a 3 or 4 day sampling trip to East Maui, and we collected nutrient, FDOM, and isotope samples, as well as a few other samples. And I'd like to use these samples along with hui o ka Waiola data to conduct a 1 week and 4 month post-storm analysis of East Maui. And then you can see me at one of my sampling sites in East Maui collecting an isotope sample, and then some of our preliminary FDOM data. Don't try and take any conclusions from that. I just wanted to tell you, we have about 300 samples left, so not quite there yet. And then acknowledgements. I just wanted to thank everyone that helped me with my project. Obviously my mentor, Dr. Kealoha, and then especially Maddie and Sam. Maddie helped me run the seal, which is the scariest and most finicky machine I've ever had to use in my life. It was terrifying. And then Sam helped me learn how to code, which was, again, one of the hardest things I've had to do in my life. I still wake up with like cold sweats thinking about like whether or not my code was wrong. So yeah, thank you to everyone that helped me with my project. Any questions?
Anybody have a question for Hannah? I don't know if we can. Oh, go ahead.
So FDOM, almost there. But curious with the isotope data, if the isotope signatures don't necessarily always look black or white, it'd be quite gray. So what kind of patterns might you expect to see in isotope data with nutrient enrichment from cesspool versus agriculture? You thought about that yet?
I don't have any prior experience with isotope data. That's something that I'm hoping to like learn and figure out. But I know that these signatures aren't always going to be black and white, which is why I'm hoping to get up with FDOM data. It's kind of look at it as a whole picture rather than just like the one. But yeah, I would have to learn more about that.
I'm not sure if we can hear people online if you unmute yourselves, or you can just, I guess, put it in the chat. You could try unmuting yourself if you have a question. If not, anybody else?
Can you hear me?
Yes, actually we can. Go ahead.
Yay. Well, that was a very well done presentation and very timely research and important, so good job. I had kind of a specific question around your salinity and N+N data. Did you need to do any kind of salinity correction for those N+N because you were gathering data from so many different salinity regimes? Or is this raw N+N uncorrected for salinity?
We did do a salinity correction. I know Dr. Stillbigger oversaw that. I'm not sure exactly what that looks like, but I do have the raw data as well as the corrected data.
Okay, so what you're showing the raw data here then in this figure?
Yes.
Interesting, cool.
Or is it corrected?
I think this is the raw data. Yes.
Okay.
Yeah, and I notice here, because you have it up, that like there's areas where there's an obvious match between like lower salinities and higher nitrogen values, nitrate, nitrite. But then there's some like Nanakuli that has like I guess you could consider a more marine or normal marine salinity, and yet you see higher N+N values. Did you have, I can't remember if you already explained that, but I was just curious if that was something that if you could have any possible hypotheses around those kinds of mismatches.
Yeah, I do have a few hypotheses, which is why I want to measure the stable isotopes and continue on with the FDOM data just to kind of see where that leads. My current hypotheses for Nanakuli specifically, we do see one more depressed salinity value paired with that one really high nitrate value. And I do know the output for Nanakuli stream is like right around there, which would explain the lower salinity and the higher nitrates. That would be my hypothesis for that specific area. And then if we look at North Shore, where we do see the lower salinity, or no, the higher salinity or the more coastal salinity along with the elevated nitrate, I was looking into agricultural runoff from the old Vailua sugar plantation, or maybe the concentration of cesspools along Haleiwa, and then something similar for South Shore with cesspools or submarine groundwater discharge.
Okay, thank you. That was Dr. Lillian Tuttle-Raz from Hilo, or
Yes.
Big Island.
Yep,
Thank you.
Hilo. Thank you.
Okay, we're going to move on now. So our next speaker is Kai. Yes, thank you.
Oh, sorry.
There we go. Now you can have it. So Kai will be a senior majoring in cell and molecular biology at Seattle University. He's been working this summer with John Burns's group at UH Hilo, learning a bit about their 3D mapping approach to evaluating coastal health. So go ahead, Kai.
Okay. Aloha, everybody. My name is Kai. I worked at the Mega Lab this summer. I'm from Hilo, Hawaii. And my project was habitat mapping of the Laehala intertidal zone using drone and underwater imagery. And I worked with Kainala Stewart at the Mega Lab. So I'm sure, as we all know, we learned a little bit about that from Hannah's presentation, that coral reefs are vital to Hawaii's marine ecosystems, our economy, and our shorelines. They also have a lot of ancestral and cultural significance. In the Kumulipo, which is the Hawaiian creation chant, the coral polyp was actually the first organism created. The first line reads, Hanauka uku ko'ako'a, hanaukana he a ko'ako'a puka, and then all following organisms came after the coral. Going into what intertidal zones are, these are at the interface of the land and the sea. So they are exposed at low tides and they're submerged at high tides. They have a very unique ecosystem because the organisms that live here have adapted to a harsh environment. They're not in water for long periods of time. The sun is super hot and the waves crash all in the intertidal zone. However, the organisms that do live here provide a lot for the community. They provide food through the limu, the opihi, and the ha'uke'uke. And in Hawaiian culture, the natural intertidal pool is called a local kaheka, which is just a tide pool basically, and they were used as like a holding zone for fish that were caught. They were temporarily put there, and then later on they were eaten. However, as climate change is accelerating worldwide, there's a lot of threats facing these local kaheka. The intertidal zones are impacted by rising sea levels because they're the first thing to impact the sea levels that rise because they're right at the interface. Also, there's a lot of invasive species that tend to prey on native species. Also on windward Hawaii Island, there's a lot of rain and the runoff pollutes the waters, which is what Hannah's thing was all about, which is really cool. And also rising ocean temperatures accelerate coral bleaching, and ocean acidification weakens CCA and organisms' calcium carbonate shells. So a lot of things happening to these intertidal zones. Going into what I worked on this summer, a lot of it was remote sensing. And what remote sensing is just collecting information about an environment without directly disturbing it. A form of remote sensing that I used was drone imagery. It's high resolution, it's geospatially referenced, it's efficient, and it's non-invasive. And we did two different types of drone imagery. We did RGB, which stands for red, green, and blue. And this is basically just visible wavelength, so what you can see with your eye. And then we also did multispectral, and that's just basically like a wider range of wavelengths that it's collecting. So it's including the visible wavelengths and also the invisible wavelengths. Another thing that I did was underwater structure for motion models. And this creates 3D models underwater of reef complexity. It's relatively fast. And we use these tools to understand how the ecosystem changes over time. And then that model on the bottom in the middle is just showing the drone is taking images at a flight plan and the images have overlap. So you can create a 3D model. Going into where we did this study, it was at the Keaukaha coastline in Hilo, Hawaii. It's not like a lot of the coast here in Hawaii, especially not here on Oʻahu. It's not sandy, it's super rocky. And the rock is basalt lava rock. There's also a lot of submarine groundwater discharge from freshwater, from the aquifers and lava tubes that discharges into the ocean. So the water is very brackish. And this brackish environment supports local i'a, which are native Hawaiian fish ponds. And they're very plentiful in Keaukaha. They supported my kupuna, or our ancestors, through sustainable aquaculture, through kilo, which means observation. So an observation of how organisms interact with each other, and pilina, which is relationship. So viewing the land not as a resource, but as an ancestor to take care of. However, as climate change and invasive species are accelerating, there's a community-led effort of restoration of Keaukaha's local i'a. This is showing the local kaheka that we did the drone imagery from. So this is drone imagery. It's called Laehala, and it's in Keaukaha in Hilo. So yeah, pretty cool. And what we were trying to wonder with this study was if multispectral imagery would improve intertidal habitat classification relative to RGB drone imagery. And we predicted that the multispectral imagery would produce higher overall classification accuracy compared to RGB imagery. And this is just because things that look similar to the eye in RGB imagery, for example, like sand and mixed rubble, perhaps the additional bands from multispectral imagery could differentiate between those better. So yeah, we predict that it'd be better than RGB. This is just showing some of the underwater reef models that we did. So this is the drone imagery, and the dots are showing where the corals models were. We used these models just to kind of ground truth what we were seeing. So making sure what we were seeing from the drone was accurate. And you can see it's very detailed. We could actually classify it by species. And it's super fast. You just go in the water, you have like a little GoPro on a stick, and you just like swim in like a lawnmower pattern. And yeah, it's super cool. Going into the drone imagery and the field work that we did, we would go out, we had the DJI Mavic 3E multispectral drone. We set it to fly at 65 meters altitude and the images to have a 70 to 80% overlap. We went out early in the morning, so there wasn't a lot of glare and reflection on the water, so we could see. And we collected both high resolution RGB and multispectral imagery. Another part of being out in the field is collecting real-time kinematic GPS points, or just RTK GPS points. It's just to ground truth the aerial imagery. Has super high precision positioning of less than 3 centimeters accuracy for longitude, latitude, and elevation. And you can see that's what that like stick is in the bottom picture. It's the RTK GPS thing. And then we also went out at extreme low tide. So we went out at king tide, so we could just see things that would usually be submerged were visible for the drone. After we were out in the field, we would go back to the mega lab and post-process everything. So we would generate the orthomosaic in Agisoft Metashape, and then we would put that orthomosaic into ArcGIS Pro, and we would perform supervised image classification using the image classification wizard tool. And then after doing that, we would do object-based image segmentation. So that's what this is over here. We would create different habitat classes. So each different color of the polygons is the different habitat classes we chose. And then we would create around 50 per habitat class. And then after that, we would do the support vector machine classifier, which would give us some validation points. And then we have to go through and make sure that what it classified was correct. And if it wasn't correct, we would have to correct it. And then after we would correct it, it would give us the confusion matrix and the accuracy percentage, along as the kappa value, the producer accuracy, and the user accuracy. And then we would do that both for the RGB imagery and the multispectral imagery. These are the different habitat classes we chose. So this is straight from the RGB drone imagery. We had rock, macroalgae, water, sand, live coral, turf algae, CCA, and mixed rubble. This is how it did for the RGB imagery. The orange is the rock, the yellow is the sand, the green is the mixed rubble, the pink is the macroalgae, the red is the CCA, the teal is the live coral, the orangish is the turf algae, and then the blue is the water. So you can see it did pretty good. And then this is the same thing, but for the multispectral imagery. So you can see that they're quite different from each other in how the classification did. And after going through the accuracy data, we found that the multispectral imagery did improve the overall classification accuracy, but only by about 10%, and it varied greatly by the different habitats we had. One of the things we looked at mostly was the producer accuracy, which is asking how much of the real habitat did we correctly classify. This graph over here, if you look at the x-axis, it's showing the habitat class, and the y-axis is showing the accuracy percentage, and then the different colors of the bars. So the blue is the RGB producer accuracy, and the red is the multispectral producer accuracy. So you can see that for sand, mixed rubble, and water, the multispectral had a higher producer accuracy. However, for macroalgae and CCA, the RGB actually had a higher producer accuracy. And then for the other habitat classes, they mostly stay the same. And then for the user accuracy, so the user accuracy is just asking if we were to look at the map and look at something that it classified as water, what is the percentage that actually is water. You can see that for both multispectral and RGB, it didn't really differ that much by habitat class, but it was low throughout besides rock and water, which had over 50% user accuracy. So this contributed to our overall accuracy difference only being about 10%. And also our change in kappa value is only about 0.1, which is also about accuracy. So yeah, after looking through, we realized that it varied a lot by habitat. The multispectral did a lot better at classifying mixed rubble, water, and sand, but the RGB performed better for macroalgae and CCA. And then the other classes showed little to no difference between RGB and multispectral. And looking at why this might be, some of the habitat classes may have overlapping spectral signatures, especially for the multispectral. The addition of those extra wavelengths may be too similar, and the classifier may be getting confused. Also, for mixed rubble, it contains other classifications like rock, sand, and algae. So it could be classifying mixed rubble as like sand when it's actually mixed rubble. And so yeah, there's a lot of benefits and tradebacks to using the multispectral. Also, the RGB color and texture, so just what we can see with our eyes, could be sufficient enough for habitats that are visually distinct, like rock and sand. Looking at our conclusions, we concluded that the high-resolution drone photogrammetry provides a repeatable, scalable, and efficient tool for monitoring these sensitive intertidal zones. And the value of using this multispectral imagery depends a lot on the habitat you're trying to classify. The RGB imagery could be effective enough for habitats that are very visually distinctive. And these tools, so remote sensing and geospatial tools, complement the Hawaiian values of Kilo and Palina. It provides coastal communities with a different way to look at their aina through a bird's eye view, and with data they can use to lead to restoration of local i'a in Keaukaha and elsewhere in Hawaii. The future work that we could do would be to redefine our habitat classification and samples. So we could possibly consolidate spectrally similar habitat classes. For example, instead of having like mixed rubble and sand, we could just do one that's mixed rubble / sand to increase our accuracy. Or we could also increase our number of training polygons to provide it with more data. We could also integrate our underwater SFM models. So we classified the corals down to the species level, but also we classified the algae and the urchins down to the species level. And this is just more data that the community could use for restoration. And then also long-term monitoring. So going out again and doing another drone survey and seeing how maybe the habitat changed over time or how the sea levels rose, and then providing that to the community. Another big part of this summer was being in the community and doing a lot of outreach. That was super cool with the Mega Lab. They're really big on that. And it made me think about how science is only beneficial if you're giving it to the communities who need it the most. We would go out and provide the community with these tools, teach them how to use it, and let them know all about it, and just extend an open arm and then provide that data that we collected for them so they could use it. We did some stuff with kids, which was kind of crazy actually, but it was fun. And yeah, that was a big part of my summer and I really enjoyed it. And I would like to say mahalo to my mentor, Kainalu, the Mega Lab, everyone in there, but especially John, Honani, and Makoa, and then also UHP Low for letting me crash all summer. Hui Hole Maluo, they're a community down in Keaukaha that restores a lot of these local i'a, and they were super welcoming to me, and it was cool seeing how they operate, and of course, Boise Grant. Mahalo.
Very nice. Thank you. Do we have any questions? Yes, here we go.
So my question with how do you achieve this with deeper habitat or habitat that's less clear? So you mentioned local i'a a bunch, but if you were in a local i'a, the water's more turbid, and it might be harder to resolve some of the things on, or if you were in another reef habitat area where it was a little bit deeper.
Yeah.
What kind of limitations are there for visualizing?
It would definitely be difficult for the drone to pick up deeper water. All the times when we were trying to build the ortho mosaic in Agisoft and like the water was deep, the photos just wouldn't align. So there'd be like, actually you can kind of see it in the coral models, like over there's like that puka in the coral because the photos didn't align. So that would probably be the same thing if the water was deeper. So I would say we could do the underwater model. So just going in with the GoPro and swimming in like a lawnmower to try to classify it. It would just take longer and we'd have to have like some really good breath strength to do that. But yeah, I don't think the drone would be as successful at capturing the deeper water because, yeah. But thank you for your question.
So for this one, you were underwater.
Yeah.
Yeah, I just want to ask that, like drone imagery versus a GoPro or like satellite imagery.
Yeah.
Scale you can to that.
Yeah, that's a great, I don't have the exact number of the resolution. I will say that we did work with satellite imagery, and then we would also go in with the GoPro, I mean, not the GoPro, the drone imagery. And the drone imagery is a lot higher resolution. You can zoom in a lot and you can still see a lot of the coral. But yeah, I don't have an exact number for you. But the SFM model, so the underwater one, is super high res, like just from looking at it, I would say. But yeah, that's a question to ask my mentor, Kainalo, and he's not here.
Okay, I think we're going to move on. Thank you very much.
Oh, thank you.
All right, next up, we've got Hikaru. Okay, so Hikaru graduated in June with a marine biology degree. No, not yet, not yet. Oh, I thought you had. Sorry, she hasn't graduated yet. Almost though, right? Yeah, okay. From UH Manoa. And she has spent the summer working in Fernanda Freitas's lab using cutting edge 3D holographic microscopy to examine some ornery plankton. So tell us about what you found.
Hi, I'm Hikaru, and this summer we were measuring the refractive index of phytoplankton across physiological states. So first off, what are phytoplankton and why are they important? They're microscopic microscopic photosynthetic organisms that live in aquatic and sunlit environments. And they're the foundation of marine food webs supporting zooplankton and thus higher trophic levels. And they're responsible for half of the global primary production, so fixing carbon into oxygen. And they're highly diverse. As you can see, these are some examples: cyanobacteria, diatoms in shape, physiology, and size. So then how do we quantify it? How do we quantify the phytoplankton in the ocean? Scientists use satellites, optical sensors on ships, and autonomous platforms to estimate their biomass globally, as well as an example. They also track chlorophyll concentration as well, as you can see on the top right. But then these measurements occur at such a bulk scale that converting them to useful metrics like carbon and biomass requires some assumptions. So for an example, when they assume that higher signals would mean more or larger particles, but those same signals could come from smaller cells with denser intracellular carbon content. And historically, it's difficult to distinguish these two, or these signals for cell size, abundance, and intracellular density. And the refractive index is a proxy for intracellular carbon density of particles. So why refractive index matters is it's a measure of how light is bent in a medium, and it's directly related to its intracellular mass density. And most biooptical models use an assumed value for it to be able to convert to biomass. And this lack of knowledge how refractive index can change through time and space might be biasing global estimates of phytoplankton. So we've long recognized how important it is, so we just don't have the technology to be able to quantify it. And some refractive index studies have already estimated that lab-grown cultured phytoplankton can range between 1.352 to 1.467. And now we have new instrumentation to be able to directly quantify refractive index using the NanoLive that I used this summer. So it's a microscope, it's able to use holographic techniques to be able to measure the refractive index directly from the particles we're looking at. and also be able to track it over time. So we're able to look at the same cell over a long period of time. As you can see, this is a time lapse that we got. This is one of our Thalassiosira. It's a type of diatom, as well as a really good image of Tetraselmis. This is what we were looking at mostly. And this is very new, so this is some images that I got. And by being able to use this instrument, we can also track single cells through their viral infection. And viruses are important because phytoplankton, under the right conditions and with enough nutrients, form these things called algal blooms that was introduced earlier. And in the open ocean, it can form so densely and on the surface of the ocean. You can see it from satellite images spanning as big as hundreds of kilometers. And viruses are important because they're able to regulate phytoplankton when these events happen by infecting them and killing them. And what the viruses do is they use the hosts to change its metabolism and intracellular structure to be able to replicate itself, to have hundreds of viruses within one cell. And we assume that this might affect the cell's refractive index. It'll change from how it normally is to after it's full of viruses. So being able to see those different signals and be able to differentiate it would allow us to use this information on the ocean. And this is a time lapse as well of being able to see some phytoplankton be lysed, just where it explodes because the cell wall is open. It's very cool. And so knowing all of this, a few questions just two that we were asking this summer. So how does viral exposure alter single cells' refractive index in phytoplankton? So we hypothesized that the refractive index would have a peak. So right as viruses multiply to the max amount, usually between, like, for our specific one is about 800 to 1,000 according to Gajigan 2025. It would peak because it's such a small area with so many viruses before declining as the cell is lysed and open to the environment. And our second question is, what would the average refractive index of green microalgae be? We hypothesized that it should remain similar between nearly identical cultures, but we really don't know that much about it and how it can change over time. So we wanted to look at that. So our methods is for viral infection experiment was that we're looking at culture Tetraselmis, and we'll have two different samples, the giant virus called TETV, Tetraselmis virus, and then the uninfected control. And we'll be looking at them, the single cells throughout their known viral infection cycle and beyond their expected lysis. So it's expected, as the study I cited earlier, Gajigan 2025, under the right conditions, it might be 16 hours, but it may change depending on how its environment is. And average green microalgae refractive index, where you comparing two separate cultures from the same order called Chlorodendrales, and we're comparing them, their difference in changes in refractive index over 80 hours. So for our results, our viral infection, on the top right is an image from A is a cell before, and then 2 hours later, it is shown lysing as its insides are coming out. I would point at it, but it's in the top right. And within just 2 hours, the cells can lyse. And this is one cell among hundreds that we were looking at. And so just to see the pattern of what happened, our maximum intensity projection, it's basically just the brightest frame for each particle over within that hour, and we were recording for 80 plus hours. And each dot represents about an average of 300 cells. And in the beginning of our graph on the right side of it, you can kind of see a sudden decline. We're not too sure what exactly caused it, but we think it's due to cells being under stress or a problem with our methodology where it just takes a little time for it to settle within the instrument. And we started with a dark period of about 48 hours. And to be able to just have the control, so it isn't being, the cells aren't active, and we could just see how the viruses multiplying inside would affect the cell. But we found out there wasn't any change going on, so we needed to expose the phytoplankton to light. Whereas then 20 hours later, then we finally saw them start to lyse, and there being changes in its refractive index over time. So then we can see a divergence between the control and then the infected Tetrasomus. So the infected one is going down. And then for comparing our two cultures, initially we thought we were comparing two Tetrasomus cultures, but just this week we found out, it turned out that all we know is down to the order, which is Chlorodendrales. So we can't say that we're comparing the same or very similar. They're not both Tetrasomus, but they're within the same order. So they're very similar in function, and we also couldn't tell the difference between them. But when you look at them under the nanolive, you can see that there's a very different difference between them. For culture 1, the refractive index is much lower and has a size that's a micron bigger than the other culture, which would mean that it has a lot more, it has a higher water content. In comparison to culture 2, it has a higher refractive index and a lower size. The diameter tells us the relative size of the plankton we're looking, or the phytoplankton, and this would mean that there's a range of 1.367 to 1.402 in just the same order, which would mean that this means other phytoplankton might have just a similar range. And then just some additional stuff, we were also looking at diatoms. So this is our Thalassiosira. We didn't have enough data on them to be able to make any conclusive things about them, but this is very interesting. We segmented the cell. We can see we're tracking their size over time. This is over 20 hours, and you can see that the brightest points are where they're packed with carbon. It's the higher refractive index, and then the darker spots are where there's a lot more water content. And this is them changing over time. And you can also see on the right, it's a little bit blurry on the screen here, but in red around the diatom in the middle is our bacteria. And we were seeing how they were multiplying around it and how it was interacting with the diatoms over time and how the organelles changed as well. And then just some takeaway points over this summer. There wasn't evidence for a peak from viral loading, but we did observe a refractive index decline as cells began to lyse, which might be an optical signal for cell lysis or viral infection. And the refractive index of green microalgae was significantly different across two similar cultures, which means it's showing the importance of characterizing refractive index across and within multiple phytoplankton groups. And understanding these changes at a single cell level can be used to apply to the bulk measurements that we had from earlier to have more accurate measurements of ocean ecosystems and biogeochemical cycling. And some future directions that we have. We also want to keep tracking, we want to track the life stages of phytoplankton to be able to record their size and refractive index as a range in similar experiments. And then we could also see how refractive index changes as cells respire through natural death and bacterial decomposition after seeing their viral infection cycle, and also compile a catalog of refractive index across taxa and consider recording how their changes in physiological state can also affect the refractive index. And then thank you so much to Fernanda and Robert this summer for helping me during this internship, and then to OIC Grant for this problem opportunity. Use up my references.
Excellent. Very cool. Thank you. Do we have any questions? I love this connecting the tiniest stuff to the global scale.
Yeah.
That's kind of amazing. Yeah, Elyse.
How are you guys able to figure out the vaccines?
They use like a, I think it's from their DNA. I'm not too sure. So there was some that was down to Tetrasomus, the genus, but mostly just overall Chlorodendrales in that culture. So we couldn't say for sure that it was Tetrasomus. Any other questions?
Anybody online have a question? Feel free to unmic or unmute. No? Oh, yeah, go ahead in here.
So
What did you learn here?
So because if we can start, well, not just with this, just phytoplankton, because we only have about 2, but if we can start making a catalog of more refractive indexes, then that means we can have those specific refractive index to be used in those bulk measurements to have more accurate readings of those signals into biomass and how much carbon there is in the ocean and how those things change over time. Since right now, they don't measure the refractive index when they're measuring these things. So it might be different at different points in time, but it's assumed to be this relatively the same.
Okay, thank you very much. We will move on. Our next speaker is Leilani. Leilani is a rising sophomore in biological engineering at UH Manoa, who worked with Camille, oh, I'm going to mess up your name, Paniello this summer. She's been helping create an affordable underwater microphone to capture the sounds of healthy coastal ecosystems. So why don't you tell us how that system worked this summer?
Yeah, thank you, Rachel. Aloha mai kako. My name is Leilani Phan, and today I'm going to be presenting my research on KaniMoana, the development of a low-cost open-source underwater acoustic recorder. So if you ever stuck your head underwater, you can kind of hear like this crack, crinkle, and pop sound. Well, that actually comes from snapping shrimp, which is one of the many things that help contribute to a coral reef soundscape. Some other things that might contribute to that can be things such as wind, rain, and even human activity. And by listening to these sounds, it offers a non-invasive way for us to better understand Hawaii's coastal ecosystems. So what exactly can these sounds tell us? They can tell us who is present, what they are doing, when they're active during day or night, and how exactly is the ecosystem changing. But how do scientists exactly do this entire process? Well, first, we have to deploy an underwater acoustic recorder that has a specialized microphone that we call a hydrophone. From there, it can pick out different sounds made from different organisms. And when we retrieve this recorder, we can take these recording files and turn them into spectrograms. So these are some examples of spectrograms. So as you can see, on the x-axis, it is time, and on the y-axis, it is frequency. And the more darker it is on the spectrogram, the more energy that call can have. And by looking at these spectrograms, we are later able to answer our questions of who is present, what they are doing, when they're active during day or night, and how is the ecosystem changing. But for the purposes of today, we're gonna focus on acoustic recorders, my favorite part. So acoustic recorders, there's many different types out there in the world. But existing acoustic recorders are not only expensive, but they're also difficult to repair. And existing low-cost alternatives have 2 main issues. The first one is that they're directional. But what exactly do I mean by the word directional? What I mean is that they only record audio from the way they're facing, whereas if you record omnidirectionally, you can record from all around. And this is especially important in an ocean soundscape where sound can basically come from anywhere. Secondly, the other issue is that they use a microphone and not a hydrophone. And what exactly is the difference between the two? Well, a microphone is specifically made to record in air, and it's made so that it only records within 1 atmosphere, whereas a hydrophone is specifically made to record underwater, and it's able to have the sensitivity of not only to like record, or like it's made to have a more sensitivity because it's under a lot more pressure compared to a microphone, because the deeper you go underwater, the more pressure it creates. And they also just have 2 entirely different sensors. I could go on and on about this, but that's like the main difference between the 2. So to address this problem, I designed KaniMoana, which has an omnidirectional hydrophone and is also modular, accessible, and affordable. But I first want to talk about the first iteration of KaniMoana, which uses off-the-shelf electronics and can last 24 hours using 14 AA batteries. When I took on KaniMoana, I had 3 main objectives. The first one is to improve operational accessibility, to create a modular battery pack to expand deployment time, and finally expand system logging. So first, the way I improved operational accessibility was I included a power switch. And I know this seems very minor, but the reason why is because before with KaniMoana, you had to turn on the electronics before sealing up the entire enclosure and then deploying it. Whereas now with the power switch, you're able to turn on KaniMoana externally, which makes it a lot easier for the users. And to make sure that KaniMoana is booted up properly, I also added an LED indicator light, which will blink for 5 seconds every recording cycle, allowing the user to know that, hey, this recorder is ready to just put in the water and deploy. From there, I also started to create a modular battery pack. So I went from AA batteries to D-cell batteries so that they last longer. And I designed and 3D printed my own custom battery holders to do this. So breaking it down, I first have a top plate with copper tape and connects all the positives. Then I have individual battery layers, which creates a 4 series 7 parallel battery. And lastly, I have a bottom plate, which also uses copper tape and connects all the negatives. And you might be wondering, well, why copper tape? That's such an interesting way to make a battery. Well, traditionally, we know that batteries are made using springs and battery tabs. However, one of my biggest constraints when building KaniMoana was the sizing. So I had to go with a unique alternative in designing my battery, which is how we got to this. And when we talk about how KaniMoana runs as a system entirely, I have 3 main boards. So the first one is a Raspberry Pi computer. Next is a Witty Pi power management board, which helps turn on and off the system. And then we have a Hi-Fi Berry sound card, which is what is used to help record the entire thing. I also have a step-up booster to regulate it at 7.5 volts so that it can last longer in the field. So when we talk about the code of KaniMoana, once you install the code, then what happens is one program will start a log and schedule when KaniMoana will record, and KaniMoana is set to record near continuously with 26 minutes recording and 4 minutes standby. From there, the actual audio recording file will start, and this is when we see that LED blink and the audio file is being generated. And by the end of your deployment, once you turn off the recorder, there will be 4 main types of files on the USB. The first one will be a KaniMoana log. The second will be a Witty Pi log. Third will be a schedule log. And lastly, all the different audio files in general. So in summary, I first want to start with the battery life of my recorder. So I'm still in the process of testing it right now. So unfortunately, I do not have a definitive answer. However, according to my calculations, we are looking at a deployment time of 2 weeks of recording. So I'm excited to see if I'm actually right. The next one is that it costs $850 USD, but including tools, it's $1,300. However, one thing I want to note is that there's only 4 specialized tools that you need, which would cost $200, but everything else is commonly found in makerspaces. Next, it has a recording cycle of 26 minutes recording, 4 minutes standby, and it has a sampling rate of 48 kilohertz. But how exactly does KaniMoana compare to other acoustic recorders out there? Well, first, we deployed KaniMoana in Palmyra back in 2022. And from here, we looked at 3 main types of recorders: Hydromoth, GoPro, and KaniMoana. These 3 are low-cost acoustic recorder alternatives that all cost under $1,000. But there's some main differences I want to highlight between them before I kind of dive deep into the data. The first is that Hydromoth and GoPro are directional and they have a microphone, whereas KaniMoana is omnidirectional and has a hydrophone, meaning that KaniMoana should be able to pick up more sounds compared to Hydromoth and GoPro. Additionally, there's differences in the battery life. Hydromoth is able to last to 240 hours, GoPro is 2 hours, and KaniMoana is still to be tested. And Hydromoth is able to have a max sampling rate of 384 kilohertz, GoPro is 48, and KaniMoana is 192. But for the purposes of testing, they're all set at 48 kilohertz. So let's get into the data. So this is a lot to take in at first, so let me break it down. On Raven Pro, when all the spectrograms are set to be the same visually, you can see that on GoPro, it's like almost basically nothing. And this makes it really hard to be able to know if GoPro actually picked up anything. And so I did have to make some visual adjustments so that we are able to see these calls on the GoPro. The first thing I want to highlight is that within these red boxes, let me break it down first. On this x-axis, we are in timestamps by 10 seconds. And on the y-axis, we are looking at 4 kilohertz frequency and below. And in this red box, we're looking between 1 to 2 kilohertz frequency approximately. And one thing I want to highlight is that KaniMoana is able to pick up an entire fish chorus within that range. Whereas with Hydromoth and GoPro, we don't really see that. And the reason why that this is important is because if we used only Hydromoth or GoPro, then we wouldn't have really known that there was these type of organisms making these sounds in the first place. And secondly, another thing I want to highlight is that on KaniMoana, not only is it able to pick up the same calls as the Hydromoth and the GoPro, but it's able to pick it up at a higher energy compared to the other ones. But this was during like a daytime deployment, and we all know that like there's not a lot of fish activity in the daytime, so it's not as exciting to look at. So my favorite part is looking at the nighttime. So unfortunately, by then the GoPro was dead, and so we're only going to be able to look at Hydromoth and KaniMoana. But with Hydromoth, as you can see, there's 3 distinct different types of fish calls. I like to call them by frog, alien, and pulse. And we're able to also see that, like we saw earlier, within this 1 to 2 kilohertz range, we aren't really able to see any sounds at all. Whereas if we look at KaniMoana, not only are we able to see the exact same calls picked up by Hydromoth on KaniMoana, but we're able to see it get picked up to like a higher frequency, and it picks up an entire fish chorus with distinct calls that we would not have known if we were using Hydromoth only in the first place. And so for my next steps, I want to continue my data analysis and do field and tank calibration on KaniMoana. And some key takeaways I want to take about KaniMoana is that it's really user-friendly, especially for those who may not have like a background in engineering. I know like I'm majoring in engineering, but I don't have as strong of a background as you might think I do. And so I think I think it's really user-friendly, and I also made my own build guide and software for people to be able to recreate their own KaniMoanas. I think one of the best things like about KaniMoana that I found really interesting in looking at all the data was that it's really shines in picking up like that mid-range frequencies. And my favorite fish call, the alien calls, KaniMoana is able to pick up a lot more consistently compared to the other 2 recorders. And I just want to thank Hawaii Sea Grant for sponsoring my surf fellowship this summer and allowing me to work on this, as well as the people in my lab who have helped me in every single obstacle that I ran into. And mahalo nui loa. Are there any questions?
Wow, that was impressive. I don't know how to make anything like that. That's very cool. Give me a screwdriver, I might be able to bolt something together, but that's about it. Actually, I'm curious, what kind of things did you have to do to take into account the fact that this is underwater, like to avoid, I don't know, interactions with the seawater, like messing with the metals and the connections and all that? Is it all sealed?
Yeah, so I used a Blue Robotics watertight enclosure, which is one of the reasons why KaniMoana is a little bit more expensive, because it was half the cost of the entire system. But Blue Robotics, it's an open source like company. So like if you have any issues, there's like a lot of resources out there for the user to be able to help with. So like because of that, I didn't have to worry too much about like making sure the enclosure was properly working, if that kind of makes sense. And I also just used a specific hydrophone that my PI has used before in the past. So yeah, I don't know if that perfectly answered your question.
Kind of. I was also curious about the depth. Like,
The
is there a depth limit to
So
how much it can take?
the watertight enclosure is rated up to 100 meters for deployment. But unfortunately, I have not tested my version of KaniMoana yet, because the main differences between the 2 is just the battery life. But other than that, the recording and everything else is the same as the Palmyra deployment.
Yeah. Beth, do you want to go first?
I was really, I think you're really good at engineering.
Oh, no.
So my question was, you don't have time, you said you don't know how long it goes, but earlier you said something about 26 minutes.
Yeah.
So, is it that you think it only records for 26 minutes, or you connect the dots there for me?
Yeah, so the program is set so that it records for 26 minutes, and then it shuts down the entire system for 4 minutes, and then it will boot back up, and that's how the cycle goes. When I said 2 weeks, it's like the approximation of like how long the battery pack can power the entire system.
Quick follow-up.
Yes.
I know that's the Hydromoth relative to your sensor. There's some spots that are hotter in the Hydromoth and cooler in yours.
Yes.
Same sort of like target areas. Like at the bottom right over there, you see
Yes.
like 1531.
Yes.
Kind of notice there's like the hotter on the left side. And so I'm just curious, that's because of factor in? So there's something else, the directionality to it, or there's just something of picking up at a frequency is better at?
Yeah, so when I was actually looking into like documentation, I found that like hydromoth is like really good at like picking up like the higher frequency parts compare like and like KaniMoana, I haven't really looked into super depth with that yet, but these are just screenshots that I've taken on Raven Pro, so I haven't actually been able to like be able to graph these on like MATLAB to make sure that's like accurate, if that makes sense, because Raven Pro is more of like a software to help visually see everything and like figure out like where the box is and like the timestamps.
Yeah,
Is that-
you have better coverage overall across the kilometers there, so it's certainly you get more of everything. I think that's just a portion that's.
Did that?
Yeah.
Okay,
That's awesome.
thank you.
Quick question. Beautiful job. Thank you so much. My question is, where did you deploy your sensors to do this?
So this was, or KaniMoana was originally deployed by Camille back in 2022 in Palmyra, which is like, as you can see here, this is like how it was
Yeah, yeah.
deployed. Unfortunately, I did not have enough time to actually be able to deploy mine. I know it was such, I'm sorry, it was such a low. I was supposed to build 3 of them actually, and then I cut my finger really deeply halfway through summer where I just could not build for like a good 2 weeks. Not in the lab, not in the lab.
Not
It
in the
was
lab.
in the it was in my own home. I was using a knife. Yes,
Okay.
yes. But
My follow-up would have been like, because this one you're in an environment where you're just
Yeah.
picking up anything that's there. So
Yeah.
we're testing, putting like actual sounds that you know you're delivering
Yes.
to see how it feels.
yes, so that would be the field calibration
Yeah.
that I'm hoping to do in the future. Aya?
One more. Yeah.
Basic presentation. I was just wondering if you've been having it in the tank since the battery life, correct?
Yeah, but then I ran into an issue, so now I'm kind of like trying to solve that issue, so I haven't been able to like fully test it yet.
Question.
When did I pull it out?
Because you said the approximation has been 2 weeks for your battery life. How long was it underwater after you took it out?
It was 2 weeks based on my math, not by actually doing it, but I had it in the tank for about roughly 5 days before I pulled it out, and then I left it out, and then I ran into this issue of I misread my LED light. So, yes.
Yeah, that's
So future tests to come.
Yes.
Okay, thank you. That was great.
Thank you.
You're good. So I had built into this a 5-minute break at this point, but everybody's kind of ticking along. Do people want a 5-minute break? Bio break? Okay, come back when that clock says 2:17, which is 4 minutes fast apparently. So really 2:13, and we will continue. Okay, so people are going to be trickling in, but let's get started so that Luke doesn't get shortchanged here. So our next presentation is by Luke, who graduated this year, right? Yes. Okay, I got that one right. A marine science major, but a sustainable agriculture minor. So that is actually playing a role in his work this summer with Ashley McGuigan's group on an agroforestry project. So let's hear all about this.
Aloha, everybody. Thank you for being here today. My name is Luke Peyerwold. I'm a CERT fellow. And I had the privilege of working with Tristan Weil, Dr. Ashley McGuigan, and Christian Kolo this summer at the Komohana Research and Extension Center. This was made possible through the Sea Grant and CERT, and supported by the U.S. Forest Service and Agricultural Research Service at the Southwest Climate Hub. So I'm going to tell you today about my experience in ʻulu agroforestry, specifically feral pig management and drought response in ʻulu agroforests. A little background on what cooperative extension is, the focus of what I was doing. It is meant as a mostly free resource to the community to address their needs and concerns. It is a reciprocal relationship where members of the community can voice concerns or interests they might have that need to be addressed, and they will work with extension agents to address those concerns and find solutions. If solutions don't already exist, extension agents will work with extension specialists to develop research and find those solutions, which they will then share back with the community. Some examples of what this can look like can be like gardening programs, youth development programs, diagnostic services, safety trainings, workshops, and publications. It looks kind of different or varies between institutions, but the overall mission is to assist and give back to the community. And that's where the idea of meeting people where they're at comes in. While developing her master's thesis, Tristan and Dr. McGuigan found that there was a knowledge gap in agroforest practitioners' perceptions and awareness of climate change. To address this knowledge gap, they developed a series of questions to gauge, to better understand that knowledge gap. So these questions related to the agroforester's background, climate problems they may have already experienced, or like disturbances, expected risks for the future, and also mitigation strategies they may have taken to address these risks. And so some of these stressors that came up were droughts associated with El Nino and feral pigs. And although these are two distinct problems, they have compounded effects on agroforest functioning. A study in Nepal found that unpredictable weather patterns like droughts or floods can reduce food availability in natural habitats, which drives wild pigs into human areas, such as agroforests. And although this is not the focal point of this research, it is important to understand because it prompts further research in Hawaii, looking at how it impacts agroforests here. Because as we see these disturbances increasing in frequency and increasing in severity as climate change worsens, we see that agroforests are threatened. The ecosystem functions and food production systems are threatened, which leaves agroforest practitioners kind of questioning how these evolving threats may continue to build and affect their systems, which can further affect how these systems sustain communities. To understand what we're actually talking about, I'll define what agroforestry is. Agroforestry is the incorporation of plants, shrubs, understory crops, and sometimes animals into a single food or non-food forest farming system. Many view it as a kind of like new wave type of agriculture opposed to like traditional monoculturing, but it has actually been practiced by indigenous communities globally for millennia. Problem is, oh wait, here in Hawaii, a common plant to find or tree to find in agroforests is ulu. Ulu, or breadfruit, is a staple crop. It is of great importance because it is a perennial starch crop opposed to something like sweet potato, which you have to replant every year. You plant this once and it produces multiple times a year for as long as, for a long time. It is also high in, it has high caloric content and it can be consumed at multiple stages of maturity. And I forgot to mention, though the history of domestication and evolution is complex, many members of the family are native to Southeast Asia, but have since been distributed throughout the equatorial belt. But it played a huge role in Austronesian expansion. And a large part of this, not just for the food, but also because the wood is used for building things like canoes, and the latex from the plant can be used as glue. That's the third picture right there. The inner bark can be used for some stuff like cordage, and the sheath of the flowers is used to make leʻamala ʻulu. In addition to its natural resource properties, it is also of great cultural significance. Here in Hawaii, it's a Kona Low, or physical embodiment of the Akua ku, and throughout its range of dispersal, it is a symbol of endurance. But the problem is, ulu agroforests are being threatened by things like climate change. El Nino is a naturally occurring climatic shift, which results in increased surface ocean temperatures affecting weather patterns. But we are approaching the threshold of a very strong or super El Nino. And so as we are approaching that, there's a greater certainty that these impacts from El Nino are going to occur. Some of these impacts include drier winters and wetter summers. What you can see in these graphs right here, a strong El Nino results in severely dry conditions. But if you were to take it one step further and look at very strong El Ninos, or what we are projected to experience, there is a notable difference in the impacts. You can see kind of a slight increase in rainfall and then a drastic dip into extremely dry conditions. And some of the risks that come from this include droughts, stress crops, and wildfires. Overall, more stress on natural resources. Another stressor to agroforests is feral pigs. These were first introduced by Polynesian voyagers as a food source, and then a different subspecies was introduced following European contact. These two populations crossbred, and there's a population boom. And kind of an idea looking forward for future research could be that if climate stressors increase, then there will be more increased pressures from pigs on human resources, such as agroforests. And that's pretty clear because we are seeing a lot of pressure, and pigs have already shown to be kind of a nuisance. They do things like damage trees through root trampling and tree girdling. They disturb soil. They create wallows to cool off and also to clean themselves of parasites. And when they're looking for grubs, they rip up the soil. There are also known vectors for many invasive species. Strawberry guava seeds can be found in their scat. The fungus associated with rapidohea death can be spread to trees via wounding. And those walls they create provide habitat for mosquitoes, which can further impact ecosystem health. And all that to say, these climate impacts to agroforestry systems are more diverse and complex than a single point stressor. And this leaves agroforester practitioners kind of unprepared for the many different ways that these things can present themselves, because we are unsure of how these effects might kind of continue to build off each other. And so a resource gap was filled during this fellowship. And what I mean when I say that is that 2 CTAR peer-reviewed extension publications will be available for public audience. These are concise, digestible, information-filled resources that are meant for public audiences. And to kind of tie it back to what I was talking about before with the interviews, is that pigs and drought were the most common concerns for farmers. And so the first publication looked at pig management for ʻulu agroforestry, looking at their risks and damages that they are already presenting, and also mitigation strategies like fencing and trapping. The other publication looks at how to immediately adapt for the approaching Super El Nino, and also using agroforestry as an overall kind of method of building resilience within an agroforest system. And so these, I had the pleasure and honor of helping co-author and co-publish these with Tristan Weil, Christian Kolo, and the folks over at Pacific Drought Knowledge Exchange. And the publication process, I learned a lot from, but overall this whole experience has really had some things stick with me. Specifically, through the interviews, being more aware of the knowledge exchange happening between farmers and agroforesters overall as a community has been really impactful, but also seeing how practitioners can work with extension services to find those solutions and kind of work together for a greater good. Like that collaboration was really impactful to see. In terms of like actually writing the publication or helping with that, it really helped me understand the importance of science communication, not just to other scientists, but to the public, because if you're not helping the community, what's the point of doing anything? And also how important interdisciplinary work is, because a lot of people can bring in perspectives from a lot of different areas, and that provides a more robust kind of way or approach of solving a problem. And I hope what I did this summer helps contribute to further collaboration, research, and mitigation, or like implementation of these ideas to help build further drought resilience. And so I would like to acknowledge and appreciate all the agroforesters and farmers on Hawaii Island that I had a chance to spend time with, to my mentors, to everyone else involved, to the professional development opportunities through SURF, and all the other collaborators that went into this. It wouldn't have been possible without everyone. So, mahalo. Any questions?
Yeah, hi.
Everything he knows well, silly, or like a lot of neighborhood. There's like a way to work them all, keep them in culture.
Yeah, well, what's great is this, these extension publications will be available for anyone, but we found that like a lot of people said fencing is one of the best ways to like overall keep them out of the whatever system you have. But individually wrapping trees was another method we talked about. But yeah, there's a number of different things we can chat more. Yeah.
I don't have a question, but I just wanted to compliment you on two things. The first was aesthetically, your slides are, I love the ʻulu in the background.
Thank you.
And then the second thing was your comment and your appreciation for doing impactful.
Thank you very much.
Any other questions? Yeah.
That some like or I also just wondered on Guam there's also a lot of feral pigs around. Some people would just like come in and eat them. Is that something that you guys have come across too, or like think that people should or shouldn't do around like maintaining this like, their own land, but also having feral pigs be around?
Yeah, I mean, I think a lot of people, or like some of the people we talked to, they'd say like, oh, we'd kill a pig and we just eat it, or like we'd share with our neighbors. But kind of on a larger scale, there's not really, we had a hard time finding like larger kind of ways of managing the pig population. So it's kind of more focused on protecting the actual agroforests that we were talking about. But like some of the easiest things are just like, one thing that attracts them into agroforest is like fallen fruit, because they can smell that like rotting stuff and that sounds tasty to them. So like clearing that and just kind of providing a line of defense. I don't know if that really answers your question, but I'd be happy to talk more if that didn't.
Okay, let's move on. Got a couple more talks. Thank you. What's this? Okay. All right. Our next talk will be by Elle. Elle is going to be graduating this fall with a marine biology major from UH Manoa. She worked with Jamie McDevitt Irwin's lab using video to evaluate fish grazing patterns. So go ahead.
Yeah, so my name is Elle, and I was really excited to be chosen as a Sea Grant Summer Undergrad Research Fellow this year. And I was working with the Jamie McDevitt Irwin lab and one of her PhD students, Natalie, to look at functional grazing dynamics of herbivorous reef fish, which I'll be chatting with you all about today. So why do we care about fish? Do we care about fish? I would say yes, personally. But fish contribute a lot to our coral reef ecosystems, and they're a huge part of how we have balance. So as we've kind of mentioned a lot today, coral reefs are really important in Hawaii for a lot of cultural, spiritual, recreational reasons. But to sort of just sum that into an easy to understand number for you, adjusted for inflation, coral reefs bring about $600 million of revenue to the main Hawaiian Islands every year. So just a really important part of the local economy. So like I said, herbivorous reef fish help contribute to the balance of these ecosystems by acting as little lawnmowers. So they graze along all the bigger and smaller algae in the reefs and help it keep it growing at a healthy level. So this prevents what we would call an algal phase shift, which is an issue a lot of our reefs are facing in climate change and other human factors, but that's basically when we see a coral dominant reef shift under stress to an algae dominant reef. And sometimes this isn't negative, but it usually is, just because we lose so many of the species that are there, as well as the ecosystem function. And then lastly, just the study, I really wanted to focus on actually the species level patterns of herbivory, as it's kind of usually seen more as like a whole with biomass or on the family level. So I wanted to see what are our actual individuals doing. So my first research question was, how do grazing rates change with temperature if they do? So other studies around the world have actually proven a pretty clear correlation between temperature and metabolic rate. So seeing a higher metabolic rate in fish as temperatures increase, which would imply maybe with climate change, we'll see higher herbivory rates with higher water temperatures. So that was something I just kind of wanted to look into. But focusing a little more on my second and third research question, and how do different functional roles contribute to grazing on coral reefs? So fish are a tad bit like people in the way that they all have their own jobs like people do. So it's important that we have somebody filling each one of these jobs. And so let's go through some of the fish jobs we'll be talking about today. So we have our scrapers and excavators, which would be like a parrotfish, kind of taking chunks. We have our grazers and croppers like surgeonfish. Browsers, so those take bigger chunks of bigger algae like sea chubs. Then micrograzers like a blenny. And finally, our territorial farmers, which are going to be damselfish. So like I said, it's important to have each one of these roles filled, but it's important to actually have what's called functional redundancy. So that's when we have multiple species and multiple individuals in each one of those roles. So if somebody dies or something happens, there's a bunch of other species to keep filling that role and making sure that job can still happen to the level it needs to for environmental health. And then my final question was looking at that specific species level. So how do distinct herbivorous species contribute to the overall grazing activity? So different species can contribute differently because they have resource specialization and different preferences. And so I kind of wanted to see if that made it so there was one species that was really a driver of the grazing and kind of carrying the workload. So the site for this study was Hanauma Bay. And if you're not familiar, Hanauma Bay is on the southeast side of Oʻahu. And it's a really unique site actually because it's very highly regulated. So there's no fishing, there's no like commercial boating, you can't take shells or anything. But it's also a really big tourist hub for snorkeling. So while there's this really high regulation, there's also a huge human footprint at the same time. And then additionally, we also were taking data on park off days. So that means there aren't any like guests allowed there. So for our actual videos, there was a little less human interaction than there might be on a regular day. So you may be wondering, how do we figure out what fish are eating? Well, we video them when they're not looking. So we would call that an in situ video observation. So we used scientific scuba diving for this project and took these weighted video frames down to kind of an appropriate habitat with cameras and let those waterproof GoPros run for about 2 hours while the divers weren't there. And that gave us the ability to kind of observe their natural grazing behavior without like us interfering. And then we chose that habitat area based on grazable substrate. So this was calculated through a benthic quadrat analysis. So we take a meter squared quadrat underwater with us just to kind of confirm that there's enough grazable substrate or like edible area for fish. And then take a photo of that area, put it into a program called CoralNet, which puts like a bunch of random points. And we could basically find an actual like ratio number of the grazable substrate using like how many points were grazable versus not. And then finally, we had the actual grazing video analysis. So for the video, we'd kind of block out that quadrat. So we're just looking at the area we have all that info on. And then taking a note of the time, family, and species every single time an herbivorous fish came in and took a bite. So kind of combining all of this, we can answer some really interesting questions. So let's answer a few of mine. So kind of bringing you all back, my first question was, how does grazing change with temperature? Does it? So just for the scope of my study, I did not find a relationship between temperature and herbivory rate. But as you can see, there's 4 months of data on this plot. So for like temperature and bite rate, and relatively that's not like a super big time scale. So there could have been a relationship observed with that if we just kind of had a little bit more data. And there is more data that just needs to be analyzed. So kind of just something to look into further. Then my second question was, how do different functional roles contribute to overall grazing on our reefs? So just this plot, our y-axis is going to be our average bite rate per minute standardized by the grazable substrate. And then our x-axis will be functional group, and then everything is color-coded by fish family. So as you can see in our grazing and cropping right there, we actually have a lot of functional redundancy in that role. But as you can see, that bar is entirely made up of the family Acanthuridae, or our surgeon fish. So we're actually getting the entirety of that functional redundancy from just one family. And then additionally, you can see that average rate is much higher for our grazers. So it's possible there's actually a serious lack of functional redundancy within our other roles. And then my final question was, how do our distinct species contribute to this overall grazing? Is there one guy that's kind of carrying the weight for everybody? So again, y-axis is our average standardized bite rate. X-axis is going to be species, and we're still color-coded by family. So I found that actually our family Acanthuridae are the drivers of herbivory in Hanauma Bay, but that driver isn't really just one species. So that's actually a good thing and kind of shows us that functional redundancy to where we lose one surgeon fish, we're not losing all of our function of grazing. But not just one species. So to kind of rope everything back in, we did not see a relationship between temperature and grazing, but that could just be this study, or it's just not a present relationship in Hanauma Bay. And then for functional roles and their contribution to grazing, there was functional redundancy within our grazers and croppers, but that was entirely made up of just family Acanthuridae. And we did see a bit of a lack of functional redundancy within other roles. And finally, how did our species contribute? Well, they actually don't just have like one driver, they contribute pretty evenly, but our Acanthuridae are really leading the charge of herbivory. So this study could go a lot more like interesting ways as well, and it is going to be. And so I'm excited to talk about some future studies and what I got to work doing this summer to kind of gather the data for those studies. So one of the things we did was stereo video analysis for fish sizing. So this is pretty much the same technique of taking those videos, but there's two cameras and this giant calibration frame, which you can imagine is fun to dive with. So we would take that to basically help us account for all the like weird angular stuff that happens underwater. Then we can use software called VidSync, and this gives us the ability to accurately size objects underwater from our videos. So this kind of adds the aspect of size and biomass into herbivory, and like, does that have an impact? How does it? So that's a really exciting thing that we're going to get to look into. We also did structure from motion modeling, which you guys got a little briefing on already. But yeah, we would take a ton of photos of the reef area we're working on in that lawnmower pattern. And then the programming Agisoft Metashape stitches all those photos together to give us an interactive 3D orthomosaic model of the reef. So this is really helpful because it gives us that data about habitat complexity, which can really impact things with herbivory. So just adds another cool aspect. And then this is really exciting to me. We did a lot of work that's going to make it possible to compare herbivory rates across different fishing protection levels. So this is just kind of a map of like all of our dive sites this summer. And as you can see, they're color coded by fishing regulation. So we were kind of all over the place and also diving in sites that have really varying protection levels. Some are like absolutely no take, like HIMB, and some are completely open to fishing. So this is just kind of something exciting and new. And we're going to get to see like if herbivory rates are different with protection levels, which can basically help inform a lot of fisheries management. So I did really want to thank Hawaii Sea Grant for making this fellowship possible. Dr. Jamie McDevitt Irwin for welcoming me into her lab. And then I wanted to thank Natalie for letting me be a part of her PhD project, and Madigan, one of the other PhD students, for really supporting me through this project and diving with us all summer. And then finally, Hanauma Bay Nature Preserve and their site curator, Ku'ule. She basically helped us have site access and gather data in a way that is respectful to the culture of Hawaii. So yeah, I'd like to open up the floor to questions.
And those are very big fish. No, those aren't fish.
Mammals.
Mammals,
Yeah.
but the prettiest. Do we have any questions in-house or online? Yeah.
Yeah, so the question was, how did we actually like annotate the fish biting in the videos? So this is really fun, and if anybody wants to volunteer to help, that'd be great. So you get to watch like every single second of all of these videos. So you'd basically put like a sheet protector over the computer video screen and like trace out that quadrat because it would eventually be like removed in the video. And then it's just like an Excel sheet, so like the exact time of like minutes and seconds in the video that bite occurred. And then usually we have good enough visibility to identify it to the species level. And then I was able to do some coding to where like the timestamps could be like adjusted to actually correlate to like a specific time of the day. That answers your question.
Anybody else? Yeah.
Of course, Kona Bay has so much like human traffic. Do you guys like integrate that into your conclusions or your data because you were able to see it while they weren't there? But like, how does that impact the fish behaviors throughout the day, if you have any?
Yeah, I think honestly that will be something we'll probably have more information on later, just because like it takes a long time to watch every single second of all those videos. So a lot of our other sites aren't really like processed yet. So I think to be able to see if herbivory in Hanauma is different from those things, we'd probably need to be able to compare it to someone else. So yeah, answer loading.
Okay. With that, we'll go on to some more fish. Thank you. I have more fish. There you go. All right, our penultimate talk is from Sarah, who is a rising junior majoring in marine science at UH Hilo. She's had a somewhat unusual summer, working jointly with Elizabeth Maiden, Lillian Tuttle Raz, and Allie here, but doing most of her work in Guam and learning how natural disasters can make coastal work a bit of a challenge. No, they underwent a typhoon, but managed to persist, which is wonderful. So go for it. Let us know what you did.
Yeah. Hi, everyone. My name is Sarah No, and I got to work this summer with PhD student Ali Malilai on her project, and we were examining coral reef halos in Guam. So just some initial background on coral reef halos. This is essentially a phenomenon that's been cataloged worldwide in all of the different oceans using in situ research and also remote sensing. There is a video. Is it working?
Try click. There you
Okay. So essentially what happens is fish, grazers, bioturbators that live within coral heads are scared by predators, and that essentially constrains the distance that they're able to graze on the substrate. And it results in these kind of blank patches around coral reefs that aren't covered in seagrass like the rest of the area is. And this also could play an important role in sedimentary carbon storage, because the farther you get from these patch reefs, the more vegetation there is and the more sediment is stored in that, or the more carbon is stored in that sediment. And just some background on Guam, so everyone knows where we got to do our research. It's a US territory in Micronesia. It's the southernmost island in the Marianas. And here there are many small fishing villages that rely on fishing as kind of a source of food, and also just for a source, or it's a source of cultural importance for them. So it's very interesting to get to research these communities because it has such an impact on those local communities. The thing that really sparked this question was a study that examined coral reef halos globally. There was a study within Cocos Lagoon in Guam using remote sensing imagery. And this here is a plot that shows that halo width to reef radius ratio over time from satellite imagery. And so we just really wanted to be able to ground truth satellite imagery and figure out whether those halos were present in both Cocos Lagoon and also other sites throughout Guam. So the overall goal of ALI's project is to determine how carbon storage capacity of Guam's coral reef halos differs across space and time, and then possibly scaling it to estimate carbon storage capacity of halo fields globally. And in order to do that and start that off this summer, we just had to determine whether remote sensing imagery accurately predicts the presence of coral reef halos in situ and characterize the biotic communities associated with these halos on Guam specifically. We used a few different methods. Before we were scouting the sites, we were actually able to just remotely decide where we wanted to go in the first place using satellite imagery and just locating potential halos on that, which I'll show in a second. And then when we were on site, we did benthic surveys with a quadrat on 20-meter transects from the center of those halos out. And we also did fish surveys both on approach and using Insta360 cameras. And the cameras were really just so that any more skittish species that would have swam away from us can still be accounted for. And we also attempted to do sediment coring using 100 milliliter plastic syringes with varying levels of success. A lot of sites ended up having just a very small layer of substrate, which just didn't really get us enough sediment to be able to quantify that carbon sedimentation or that carbon storage in the sediment. So this is just what our scouting looked like. This is on Google Earth. We were just dropping pins, pH for potential halo. And that's just what that looks like on the satellite imagery. It can be very helpful for identifying those. And this was in Hikaru Bay and Family Beach. Today I'm going to mostly be talking about Cocos Lagoon and Hikaru Bay because that's where we found our best halos. And here's just some pictures in the field. Reeling up a transect after a quadrat survey. This is on a boat in Cocos Lagoon, and then this is also in Cocos Lagoon. We were able to get a sediment core at this site, so that's what I'm holding there. And this is just a video of what that Insta360 footage looks like. So you can just see fish. We can spin it around so we can see everything and be able to see their behavior and everything that they're doing when we're not there. And in the next few slides, I'm going to be talking a lot about these three seagrass species. These are all three seagrass species on Guam. We have Inhalus acarides, which is a very large tape grass. It's usually about 70 centimeters to a meter in length, very dense. And then there are two smaller ones that grow. There's Halodula uninervis, which is only about 5 centimeters in length, a lot thinner. And then Halophila minor, which is a very small spoon grass. It just looks like little leaves, and they're only about a centimeter in most cases. So we ended up finding 3 different types of halos. We found coral halos and seagrass halos, and then we found 2 different kind of archetypes of coral halos with different seagrass in the background. So this here is just comparing in Hikaru Bay alone, a coral halo versus a seagrass halo. So in the center of the coral halo, we have these Peyerwold heads that are mounding. You can see them here. There were 5 in this halo. And then around the outside, you can see that seagrass that's growing. And the background vegetation here specifically was H. uninervis and H. minor, those smaller seagrasses. And in the center of the seagrass halos, it was actually a dense bed of Inhalus acarides instead of a coral, which you can see it's just a lot darker, but it does still have that defined halo around it. And the background vegetation was the same as these coral halos. It was H. uninervis and H. minor. And there was a little bit of Padina algae, which is that kind of flat brown algae. And this is just what that looked like while we were on the sites. This here is the H. uninervis and H. minor growing pretty far out from one of the halos. And then this is the edge of one of those seagrass halos. You can see that dense Inhalus on the inside, a bare ring of sand. There is some Padina growing there, but it's just a lot less dense the closer you get to that seagrass bed. And then this is a comparison between the coral halos we found in Hikaru Bay versus in Cocos Lagoon. Once again, this is that nice halo in Hikaru Bay with the Peyerwold heads. And then in Cocos Lagoon, we also had halos with Peyerwold heads, but the background vegetation was almost entirely Inhalus acarides, which was interesting to see because this was very prevalent in the southern area, and we just did not see that in the background at all in Hikaru. We only saw those patches. And so yeah, once again, this picture and comparing to, we had very low tides. So this is actually kind of nice though, because you can see this halo from above the water. There's that Porites head in the center, bare sand around it, and then some very thick inhalus beds off to the side of that. And then from our fish surveys, this is taken from 9 different transect surveys and on approach surveys. We haven't been able to go through the footage yet, the video footage, because there were 5 cameras and it was all very long. But there are 3 species here, the 2 goatfish and the emperor, and these are all invertebrate eaters, and they are bioturbators that disturb the substrate, which can help contribute to the formation of these halos. So we found those in 4 out of 9 surveys, 7 out of 9 surveys, and 7 out of 9 surveys, which is quite a bit of them. And then over here, extremely prevalent in all of our surveys was Halichorus trimaculatus, which is a 3-spot wrasse. And we did find this in all 9 of those surveys, juveniles and adults, and they typically shadow behind the goatfish and kind of scavenge what they've scrounged up from that seafloor. And then in 6 out of 9 of these surveys, we also found Stegastes nigricans, which is one of those farming damselfish. And these are really interesting because they do eat algae, but they are territorial with their specific area, and they'll scare other fish away from their specific territory, which kind of forces them to graze in other locations and contribute to that more widespread pattern. This is some very preliminary data. We just finished gathering a lot of our data. As you can see, the sample size is a little smaller. But the benthic cover is on here. We have seagrasses, algae, and then sediment and other. And we have these 2 different types of coral halos on the left columns there. And as you can see, in the Cocos halos, it's only Inhalus acroides. There's no uninervus or minor there. Whereas in Hagåtña, it's the opposite, and we have all of that seagrass cover with the H uninervus and H minor. And then algae, the only halo that algae was prevalent in was the Hagåtña seagrass halos. And then we found turf algae at both of those Hagåtña halos. And then obviously sand at all of them, because that's part of the deal there. And then rubble in Hagåtña coral halos as well. That's the only place where there was a lot of rubble, we noticed. And overall, this means that halo formations aren't actually limited just to live coral patch reefs. It's basically anywhere that can give a structure for fish to hide in. We were able to find it with seagrass, dead coral heads, and live coral heads. And the mechanism for formation could be the same due to similarities in the fish assemblages found at all 3 of these, but there could be other factors driving that, including just what the substrate is like, because different benthos supports different algae and seagrass and their growth there. We also found some differences in benthic cover by depth. We didn't record much of it, but at certain sites, the lower depths were a lot more bare, and at higher, just like ridges in that sand, we would find more seagrass growing there. There was also some current driving some of it, we believe, because there were certain coral heads where we would find a kind of semi-halo around it, and within these different patch reefs, all in the same location, they were all found on the same side, like going away from where the water was coming from in most cases. And then we also found that within the seagrass beds, it's possible that competition between the seagrasses and different algae could be contributing to that blank area. And we'd have to look into that a lot more, but that could be interesting. So in the future, it would be nice if we could possibly test a different coring method so that we can get back to that original carbon sequestration question. We can't test how much carbon is in the sediment unless we're able to get more significant cores. It would also be really cool if we could expand it up to the Northern Marianas. We know for a fact that there are seagrass beds on Saipan, and looking at the satellite imagery, there are also potential halos there that would be interesting to look at. And while we were there, obviously there was a typhoon, but it got us thinking that it could be interesting to see how typhoons would affect the presence and absence of those seagrass beds, because in that satellite imagery, we did see fluctuations in their size and just overall existence. So that could be another interesting thing to look at. And I just wanted to say a big thank you to everyone on this slide. Thank you to Hawaii Sea Grant and also the Hilo Student Travel Award. I had to go far. And then, yeah, people both here and on Guam helped us out a lot. So big thank you to all of those people.
All right. Do we have any questions in-house or online? Yeah.
Okay, so the question was, we were using Google Earth to scout those sites, and they wanted to know how accurate that was and what time frame those pictures are updated in. If you look at Google Earth, it's kind of misleading because it says copyright 2026, but then the image will be from like 2018, and it kind of varies depending on where you're going. So we definitely saw that a lot happening where there was an inhalus bed on the imagery, and then when we got there, there wouldn't be, or something like that. And yeah, so it's just different parts of the island, I believe, were also updated kind of at varying times. So that was part of the challenge for sure. But
Elyse.
so that answers your question.
Do we have an online question? Nope. Okay. All right, well, let's move on to our last talk. Thank you. All right. So our final talk today is from Elyse, who is a rising senior in the Global Environmental Science program at UH Manoa. Elyse is interested in coral reef ecosystem health, and for her summer project, she worked with Melissa Melendez on an experimental way to tackle ocean acidification in reef settings. So let us hear about that.
Hi guys, thank you so much for making it to the end. I appreciate you guys for being here with me today. For my project with SURF and Sea Grant, I was testing reef adaptation strategies in the face of climate change, specifically on the effects of alkalinity exposure on crustose coralline algae in Kāneʻohe Bay. I worked under Dr. Melissa Melendez and Connell McNichol at Coconut Island in Kaneohe. Just to kind of situate ourselves, Coconut Island is going to be right in Kaneohe Bay. Our point lab's right over here in the corner. I got to take a boat to work every day, which was really cool. As a lot of my fellows have also mentioned, coral reefs are super essential to overall ecosystem health in Hawaii. Hawaii has home to 85% of the United States coral reefs. They support food systems, coastal protection, culture, and tourism. And the U.S. Geological Surveys estimate about $836 million per year in shoreline protection alone from coral reefs. For my specific research, I focused on crustose coralline algae, or CCA. CCA acts as a reef engineer as it cements coral rubble together, deposits high magnesium calcite, which is the most soluble biogenic carbonate in the ocean. CCA also produces chemical settlement cues, which will tell coral larvae where to basically breed and to set, as well as accelerate reef recovery from storms and other things. And they are highly acidification sensitive, and they are a key indicator of reef health because of their high magnesium calcite skeletons. The species I'm specifically working with is going to be in the bottom right. It is going to be Hydrolithian reinbaldi. As global temperatures rise, the threat of surpassing the 2 degrees Celsius threshold agreed to in the Paris Agreement is a major international concern. With an increase in CO2 emissions in the atmosphere, humans are amplifying the natural greenhouse gas effect and exacerbate the carbon cycle, causing excess heat and CO2 to be dissolved in the ocean, leading to sea levels rising, pH dropping, which is also known as ocean acidification, which directly threatens calcifying organisms like CCA and coral. So what do we do about that? A proposed mitigation solution is ocean alkalinity enhancement. Ocean alkalinity is the measure of water's ability to resist changes in pH, so basically the ocean's buffer. Adding alkalinity substances to seawater accelerates the ocean's natural carbon sink. These alkalinity livestocks convert dissolved inorganic CO2 and seawater into bicarbonates and carbonates, which are stable forms of carbon with a lifetime of approximately 10,000 years. This process occurs naturally, but OAE accelerates this natural weathering process to theoretically increase CO2 sequestration in the ocean. So for my project, we wanted to look at how do CCA respond to elevated TA over a 30-day period? Do all alkalinity livestock have the same impact? And what is the response of CCA under different alkalinity concentrations using NaOH? So when carbonate saturation is raised locally, we hypothesize it will make it easier for organisms like CCA to calcify. So our methodology is we tested 3 alkalinity livestock that represent very different real-world deployment pathways for ocean alkalinity enhancement. Olivine, which is why the beach on Big Island is green, is a naturally occurring silicate mineral that reacts with CO2 to release magnesium, bicarbonate, and salicylic acid, but it dissolves slowly. Our second choice was calcium oxide, which is a human-made mineral, but it releases calcium and bicarbonate. And our third solution was NaOH, which is sodium hydroxide, which is a pure alkalinity livestock, which is basically just a strong base. For our target species, we use Hydrolithium rheumboldi, which are free-living marine red algae nodules that form hard calcium carbonate rather than an attached reef-building crust. For our experiment, 1 nodule fragment was placed on coral plugs and exposed in each tank. So we took a nodule, we'd use a coral cutter, and then we'd use coral glue to place it on these coral plugs. So we had 2 experiment setups over our SURF internship. For the first experiment, we had 24 coral communities with continuous flow over 30 days. In each tank, in summation, there was 144 individual coral communities, 24 CCA nodules, the 4 treatments, so olivine, NaOH, calcium oxide, and ambient seawater. And daily, we took total alkalinity measurements, pH, salinity, dissolved oxygen to ensure precise dosing every single day. That was what I got to do every day. So I got to know these coral communities quite well. There is a video here, but I don't think it will show, so I can describe it to you. But we actually collaborated with the Coral Resilience Lab. They have a proprietary software where it's basically like you put coral on stage and it gets to get photographed 244 times, and it turns on this little axle, and they're able to geometrically calculate growth over the 30 days. So we scanned our CCA before and after. We're still working on combining those images, and we did some math to find the calcification rates that I will talk about, but there was a little video here to show you how it turns. But just imagine it. Imagine it. Oh my gosh, here it is.
Video though.
Oh, that's okay. So that's what it looks like, and basically it just turns around. Okay, awesome. So to measure short-term calcification, we ran 4-hour closed incubations, sealing 12 of our CCA fragments in 250 milliliter beakers for 4 hours. Each sample size, each treatment had a sample size of 3, so 3 NaOH, 3 olivine, 3 calcium oxide, and 3 control. We let them float in our tanks, right next to our tanks, but just to isolate the net calcification, we put them on with their little floaties, and we kept them in there for 3 hours. This bar graph shows mean net calcification across our 4 treatments: control, COA, NaOH, and olivine, all at 250 micromoles, except for the control, which was at 0 micromoles. NaOH shows the highest numeric mean at about 0.12, compared to roughly 0.107 or 0.08 for the other 3 groups. However, when I ran a one-way ANOVA across all 4 groups, found that it was not statistically significant because it was over the threshold of 0.05. However, the NaOH was still found to be worth investigating in a larger sample size, and calcium oxide and olivine were not significantly different than the control. For our second experiment, we I'm sorry, guys. So based on our NaOH signal, our second experiment zoomed in specifically on sodium hydroxide, testing a dose gradient. We again ran 24 coral communities over 30 days, same amount of coral colonies. However, we used 2 different species for our CCA. So we used the same from the first, the Hydrolithian Riamboldi, as well as 24 articulated CCA species. So the articulated is going to be on the right, and the Hydrolithian, instead of cutting the nodules, we used full nodules. The 4 treatments were going to be 250 micromole, 500 micromole, 500 micromole with the additional CO2 and then ambient seawater. And I did the same daily monitoring on this experiment as well. For our second incubation, we followed the same closed chamber approach, made with some improvements. We wanted to test photosynthesis on the first experiment, but however, there was gas exchange between the oxygen within the chamber. So we tried to decrease oxygen as much as possible. And because there were the articulated and the non-articulated, the articulated were a little bit bigger, so we had to increase our beaker size, tried to make our floaties work, a little bit of trial and error to ensure almost minimal gas exchange. This incubation we did for 3 hours instead of 4, but we logged the same temperature, dissolved oxygen, salinity, pH, and TA at the start and end of each 3-hour incubation. This slide shows both response variables side by side. So this is the net photosynthesis. I'm sorry, this is the net photosynthesis. This is the net calcification split by CCA species. On the left, we have the net calcification measured in micromole of calcium carbonate per gram per hour. And on the right, net photosynthesis measured in micromole per gram per hour. In both, blue represents articulated CCA, and in orange represents the non-articulated Hydrolithian Riamboldi. For calcification, both CCA species peak at the concentration of 250 micromole and decline at 500. However, neither one-way ANOVA reached significance. In fourth photosynthesis, the pattern is more dramatic, especially in Hydrolithian, which jumps from about 1.4 at ambient seawater to over 3.4 at 250 micromole. I also wanted to compare net photosynthesis against net calcification. For articulated CCA, it stays relatively flat, mostly between 0.06 and 0.08, even as photosynthesis varies across treatments. This tells us that those 2 processes are weakly coupled in this CCA species, except at the highest photosynthesis values, where we do see calcification pull upward. This is for the non-articulated compared to the last slide. The relationship is tighter. In 3 out of 4 treatments, the highest photosynthesis values line up directly with the highest calcification, except for the 500 micromolar treatment, where calcification stays flat even as photosynthesis rises. Overall, Hydrolithian Riamboldi, the non-articulated, shows a stronger coupling between these 2 processes and higher absolute net photosynthesis than the articulated form. To summarize, both CCA species peaked in net calcification at the same dose of 250 micromolar, and both declined at 500. Though there was a weak coupling for the articulated CCA between net calcification and net photosynthesis. However, for the non-articulated, there was a higher coupling between these 2. But with both of my ANOVAs, with all 5 of my ANOVA testings, I did not find any of my data to be statistically significant. And I think that just has to do with a small sample size. Here is basically all of my ANOVA tests. As you can see, this is the threshold. And I do think that this is just due to having a sample size of 3. So to wrap this up a little bit, to scale up my replication would mean a larger target size, so about 8 to 10 per treatment. That is almost, triple what I had been doing. However, we can see that our dose response results tell us that ocean alkalinity enhancement is not a uniform fix. Higher concentrations do not mean better outcomes. And finally, before this kind of intervention is scaled up in real-world applications, we need more research and funding directed at understanding the full range and scope of environmental and social outcomes of marine carbon dioxide removal. And I just want to thank everyone at the Point Lab, Melissa, Connell, Elle, David, Zoe, Mario, and Chris for all your hard work and support. Thank you to Rachel. At the beginning and end of our experiments, we have a 26-hour experiment, so you get to spend a lot of time with your team. And I'm really grateful that they welcomed me into HIMB and helped me understand so much about CCA, because this was kind of all new to me. So I'm just super happy to be here. Thank you.
Okay. This is the problem with coral stuff. It's always hard to get results. I've seen other studies like that where you do all these careful measurements and then it's like not quite telling you anything. It's hard. They're critters.
They're doing their thing.
They're doing their thing.
It was really cool. And
Yeah.
now like,
But there's stuff to do.
and now I can notice them, and I know what kind, when I go out and I'm like, I went scuba diving, I was like, oh, that's that kind of CCA. That's that kind of CCA. So
Yeah. And there's more work to do. There's always more work to do. Does anybody have any specific questions on this? Yeah.
yes.
So based on what you found, I know your sample size limited, there are trends. If there was one treatment, this really outperforms others. And so if you were trying to take this manager or go to page or something, so you need a bunch of money to fix this problem, which one of these treatments do you think is most effective at increasing calcification of the base of ocean?
Definitely NaOH. Across both experiments, it proved to have the highest calcification rates. And that's kind of why for the second experiment, we chose to just focus on the dosage of NaOH rather than try and work with other livestock. But it seemed even in the second experiment, even though there were not totally significant, it seemed that across the board, the 250 micromoles did lend to higher calcification rates. So I would work with that livestock, probably increase my sample size, do it a couple times, maybe work with other kinds of CCA. And then I'd say, hey, Bill Gates, do you have some money for me?
Sounds like a plan. All right. Well, if there are no more questions, then I would just like to say thank you to all of the students. I'm always blown away at these events, the final symposium, to see just how much information you have picked up during the summer, because many of you, this is completely new topics. And so like plunging in and just learning so much, it's so impressive to see. So congratulations to all of you on that. And for working so hard for 10 weeks. I know that that's tricky too, because it's summer and you want to go play, but we appreciate it. And we wish you the best of luck as you move on. Please keep in touch. You will occasionally get emails from me saying, hey, alumni, will you? So just be sure to keep in touch. Let us know if your emails change and that kind of thing, because we appreciate you being part of our Ohana now. And a big thank you again to all of the mentors for being willing to step up and host these students, because as I said before, we couldn't do it without you. And tell all of your friends, all of your faculty friends, how great it was having a surf student for the summer, so that they will come and volunteer next year, because the more the merrier. Thank you all for coming online and in person, and aloha.